Hi Year 8s, just a little video here just to talk you through the microscopes we're going to be using today and over the next couple of lessons. You should be able to have one microscope per person for these lessons. So what we're going to do is go through what the parts of these microscopes are called, maybe a little bit about what they're for, and then the procedure for looking at slides properly under the microscope and be able to focus your image. All right, so what we'll start with, you come with a plug and a lead, you should have. On the diagram that you get, there's a mirror at the bottom. So the mirror is for reflecting a light source. These microscopes have got an electronic light source. I'm going to plug this in and turn the light on. So you can see here, there's a little dial that turns the light on and off. Temperamental, okay, the plug was a bit temperamental, so that's why it wasn't turning on. So the light, actually you can adjust the light as well, or the mirror underneath here, to open or close the aperture to let more or less light through. So sometimes some images are a bit too bright and you can't see the cells or the tissue or the organ underneath because it's too bright, and then again the other way because it's too dark. So you can alter the amount of light. And these particular microscopes, you can actually also, I'll turn it round, you can move the light focus up or down as well. But what we could do is just move it so that this is midway, and then move it also so that the light's gone off again. There it is. So you're sort of aperture half open. All right, so we've got not too much light, not too little, and we can go from there. So there's your light. Power on, light on. So labeling all the parts of the microscope. So we'll start from the top then. This is where you put your eye. So we call this the eyepiece lens. This is normally 10 times magnification. Okay, we come down to this part here. We've got 3 different lenses here. We call these the objective lenses. And you have the small one, which I want you to always start using the small one, which is 4 times magnification. And you can swivel this round. You should see the number 10 on the yellow one and number 40 on the blue one, if you've all got this kind of microscope. Okay, so switch that round. We've all got on the 4 times magnification setting on the objective lens. All right, now where we put our slide is called the stage, because that's where all the acting takes place, I guess. That's why they decided to call it stage, and that's where you put your slide. So what I would normally do when we're starting is move you can see this wheel here, the large focusing knob or wheel, move that all the way down to the bottom. Okay, we'll start there. There is another wheel there, you can see this one. This is called the fine focusing wheel, and it just helps you focus, make those small adjustments to focus when you're looking at your specimen. Okay, once we have, okay, we've got the handle is it's quite important to be able to carry the microscope safely, so we're using this as the handle, and actually this section here will be called the tube. Okay, here's our sample. I'm going to use, to get a good idea of organs, tissues and cells, monocot and dicot leaf transection. So we'll look at the organ being the leaf, some of the tissues being the epidermis, the mesophyll, etc. Okay, so I'm going to slip that in there and clip it using this clip which holds it in place. Now if you've got this kind of microscope, you can actually use these to move the stage around so you can find your specimen while you're looking down the eyepiece. All right, so I think we've covered all the parts there. Go through again. eyepiece lens, tube, handle, objective lenses, stage, large focusing wheel, small focusing wheel, light, well that's where your mirror would be on the diagram, power source, and that's kind of all we need to know for this video. Okay, I'm going to stop there.